So we're doing a number of things to help get get resources in your hands to help you learn as much biochemistry as you want to learn between now and then. So there should be posted on the icon a copy of last year's first hour exam. So that's one resource. Another is I have posted a number of supplementary materials that will take you in more depth on various topics that we've talked about so far. Those are available in icon under the supplementary articles. Eli and AJ have very kindly agreed to lead a review session, and that review session will be Thursday, the 12th. So it'll be before the last lecture that'll be on the test, but it'll be after almost all of them. And if you're reading the chapter ahead, you'll have some idea of what material might be still a puzzle for you. 6 to 7, Odd 3, or Watsky in Bowen Science Building. Strange names for the room. Sorry about that. Any other general questions about preparing for the exam? Yes? I think you're planning on having some presentation and then open to question and answer. So there'll be some summary and then as well open question and answer. Yes? If it's in Watsky, probably not. We can try to have it recorded, but the recorder is set up here, and it may not be possible to get it to set up there. So if you want to get to the review session, the safest way is to get to the review session. Other questions?
Is there information on supplementary materials about the text or the notes that should be on the test?
Well, anything I've mentioned in lecture or that's in the handouts that I give you for the chapters, I consider reasonable material. That's why I talked about it, is I think it's worthwhile. The supplementary material wouldn't be part of anything beyond what I say in lecture, wouldn't be on the main part of the questions. It might possibly contribute to bonus questions, but it wouldn't be the first priority for that. Okay. I'm going to be saying more about the specific details of how we're going to run the exams on Monday. So, keep your heads open for that. It'll be a little bit odd because we're going to be split between 2 rooms, and I'll ask for your cooperation in making that go as smoothly as possible. Any other general questions on background and so on? Okay. So we can close that puppy. I'll save that for later. And so we started talking about, where are we here? No. We started talking about, come on, open. There we go. We started talking about proteins and the way in which the linear polypeptide could give rise to secondary structures and to tertiary structures. Do people have any questions about the material that we've discussed so far? Okay, and I think we were here. Okay. These diagrams are not in your textbook. These are did I trash something? Oops. Oh dear. Restart it. Will it'll save all the rest of it together or what? Okay. It's okay now? All right. Hopefully that'll be okay. All right. So we'll go back to here. There's a tendency to talk about things as being hydrophobic or hydrophilic as if it's a binary, black and white, utterly and completely separate different things. And one of the principal things I'd like you to take away from these scales is showing you that there are degrees of hydrophobicity and hydrophilicity. And you can see, I think, looking at this, that there are some things which should not surprise anybody as being very strongly hydrophilic. So here's the charged amino acids, aspartate and glutamate and lysine and arginine, histidine. And these guys, especially the ones that are carrying full negative charges, are very hydrophilic. They tend very much not to sort into octanol, octanol being a fairly nonpolar solvent. Whereas the three most hydrophobic ones here are these three aromatic ones. You've got tryptophan, you've got phenylalanine, you've got tyrosine that are very strongly hydrophobic. But the other ones are more of a little bit of a shading. There's some sort of flavors of gray, if you like. And it should bring home, I think, that there is a degree to which these things happen. And I tried to emphasize when we looked at the structures of the amino acids that they differed in size, for instance, and that the bigger, bulkier aromatic ones are, not surprisingly, the most hydrophobic. And these little ones in here, although methionine and cysteine and glycine and valine are hydrophobic, they're not nearly as hydrophobic as the great big ones. Although glutamine and asparagine are polar, they're not nearly as hydrophilic as the big charged ones, and so on. This is looking at the tendency to sort out between the interface of a polar and nonpolar solvent. This is looking at the tendency to sort into octanol as the solvent overall. And you can see that this heightens the tendency for the charged amino acids not to enter the solvent, whereas in the context of the interface scale, the peptide can sort of lie at that interface, and it's just trying to deal with the side chain itself. So, okay. This is trying to point out to you the locations of the hydrophobic and the hydrophilic amino acids, showing that hydrophilic side chains tend to sort themselves out at the surface of a protein, whereas the hydrophobic ones are going to tend to try to pack away from exposure to the water, and many of the hydrophobic amino acids, in fact, are completely buried underneath that surface. Okay. So when we look at protein folding, the major source of the energy to drive protein folding is the hydrophobic effect, the fact that nonpolar things tend to end up next to each other because there's overall more disorder, more entropy from that. You might think, since there are hydrogen bonds up and down the peptide backbone, which account for things like the alpha helix or the beta-pleated sheet, you might think that those are the driving force. But you'd be wrong. And the reason you'd be wrong is that if the peptide is unfolded and those donors and recipients are in contact with the water, they can make hydrogen bonds to the water. And then when they roll up and make a cute little coil of an alpha helix, there may be the same number of hydrogen bonds, but now they're to each other rather than to the water molecules. So there isn't a big energy difference to be derived from the hydrogen bonding itself. The hydrogen bonding can give a pattern, but the overall compactness versus extended is driven more by the hydrophobic effect. We also see that disulfide bridges are also not necessarily a strong driving force. They're formed after the fact. The protein has to be folded before they're going to form. They may help protect proteins from nasty environments like the stomach and stuff, but in the initial folding process, they're not present, so they're not driving it. Ion pairs, so in some proteins, there'll be a full negative and a full positive charge against each other. Those ion pairs, again, they would have interacted with water if they weren't interacting with each other. This is giving you kind of a schematic of a comparison of an unfolded or a misfolded protein and its more compact one. So if you imagine, one of the reasons I like this diagram is that it's using something other than red-green. Red-green color blindness affects several percent of the population, and so all the diagrams that use red and green for works and doesn't work are essentially useless to people who are red-green colorblind. Blue-yellow colorblind exists, but it's a lot less common. So in this case, what we're looking at, the blue is here hydrophilic. It likes to interact with water. The yellow is hydrophobic, like butter, And in the unfolded state, you can see there are a lot of these hydrophobic residues that are in contact with the water, and that through the hydrophobic effect, will be at a higher entropy if they can get away from the water. And from that extended pattern, there might be a number of different ways to pack up. And they're showing here just as examples. Here's a misfolded one, and here is a properly folded one. In the properly folded one, you can see that the hydrophobic yellow residues have all gotten together in the center where they're not going to have to disturb the structure of water and therefore minimize the energy. Here's one where it just sort of fell into an energy local minimum or something, and it's not the correct faithful one. It's got some aspects of it. You can see there's a few aspects that are similar, but it's not quite properly folded. And in a misfolded protein like this, it would tend to aggregate out of solution. That these areas that are exposed here that have hydrophobic things still exposed, those would tend to aggregate and form multi-protein complexes that are not functional. Either of these things, either the unfolded or the misfolded ones, are not going to be functional yet. The true functionality of a protein comes from its overall folded tertiary structure. The primary sequence alone can't do it. The secondary structures won't do it by themselves. It's the tertiary structure that allows a protein to perform a function. One of the other interesting things is that these extended or misfolded things that are exposing hydrophobic stuff out where people can see it are likely to interact with things called chaperones. And we'll be talking about chaperones more later in the semester. But it turns out that there are some proteins that really can't fold by themselves. They need help, and the chaperones help with that. Whereas once they've reached its compact state like this, that's more likely to be stable. It won't be as likely to be degraded by proteases, and it's more likely to be soluble and to be able to perform the function. There are some proteins in which the zinc ion is present, and the zinc actually does contribute to the folding of the structure and the stability of the structure. Protein folding is really kind of an amazing thing when we think about it. And in the early days, it came almost as a shock that you could do some of these things. So for example, you could start with a protein that not merely had a compact tertiary structure, but that had inside of it disulfide bridges between parts of the peptide backbone. And this compact folded state could be unfolded and completely liberated from those bonds. The urea would unfold it, and mercaptoethanol would break the disulfide bridges until you had a complete unfolded state that had millions of possible rearrangements as this spaghetti-like thing kind of tumbled around. And the amazing thing was that for some of these proteins, you know, as I said, for some of these proteins, you could take this unfolded state and remove the urea that was competing for the hydrogen bonds and remove the mercaptoethanol that was covering up the disulfide bridges and be, you know, nice to it, and it would refold, and you would get back something that was the same structure and that had the same catalytic activity. And so this ability to unfold and then refold a peptide was the first strong indication that the sequence, the primary sequence of a protein, contained the information necessary for the tertiary structure. When you think about all the number of residues in a protein, and you think about each of those bonds twisting and turning, you start to realize the extraordinary number of possible mistaken conformations that protein is in. And it first boggled people's minds to think of how it could ever fold in any finite time, much less in a matter of milliseconds or seconds. And what we've come to appreciate as we learn more about protein folding, we learn more about the structures and stuff, is that it isn't that the entire universe is explored all the time from beginning to end. But if you start off with an ensemble of a very large number of things, a few of those are going to be closer to the correct state. And then that's the new population that you're working from, and so on. And so from a completely disordered gemisch like this that you start off with, as the thing is exploring all the possible states, there's an intermediate range of structures that are called a molten globule, where it's pretty compact, and most of those hydrophobic residues are somewhere on the inside, but it's not really gotten to the correct tertiary structure yet. But it's a lot closer to it, and so it's a much smaller universe of possible shapes that has to be explored. So it's not this infinite problem that it seemed to be initially. And then as you start to resolve within that molten globule further, you have a few local possibilities that have to be sorted out and explored, and maybe the chaperone will help you with them. Any questions about that before I move on? And if I've botched that completely, Eli, let me know. His lab does things like look at the actual angstrom displacement of things, so he knows this in quite a lot of detail. All right. One of the other things we've come to appreciate, which is kind of amazing, is that as a protein is being translated off of a ribosome, we now know for a fact that some of that folding process actually starts as the polypeptide is coming out of the ribosome, sort of like a crocheting machine or something. So this is showing you a representation of a ribosome reading along a messenger RNA, and this peptide chain that has been completed and is being sort of extruded out of the ribosome, there's no reason it wouldn't start folding. And in fact, we now have actual experimental evidence that for some proteins, in fact, it does start folding and does start exploring some of the native aspects of a structure. These are just some examples of ion pairs, and that they may well be present in the final structure, and them being in the right spot is an aspect of it being the right structure, not something else, but they're not themselves directing the folding process. Here's an example of some disulfide bridges, which they've cleverly put into yellow for sulfur. And these are especially common outside of cells. Most of the proteins inside the cell do not have disulfide bridges. It's more of a characteristic of extracellular proteins. And it's possible that that reflects the fact that outside the cell is a tougher place to live than inside the cell. Zinc fingers are a particular kind of local secondary structure, if you like, where the zinc ion is sitting in the middle of a domain. And it sits there in a particular oxidation state. It's not going to become oxidized or reduced while it's there. It just stays in there. And one of the things that is nifty about a zinc ion is that it can make up to 6, anywhere from 4 to 6 ligands with areas of the protein. So it's capable of interacting more widely across the protein than an ordinary side chain, for instance, might be. And that may be part of why we often find them in DNA-binding proteins. This is a structural strategy that is used multiple times in DNA-binding proteins. So I would hope, so I hope you've carried away by now that the folding of a protein is intrinsic to the sequence, the primary structure of a protein. We cannot yet predict what that final tertiary structure will be. We have good reason to believe it's locked inside that sequence. We cannot yet predict. If you tell me, a 400 residue sequence, I cannot tell you the final tertiary structure. The local secondary structures are going to form through hydrogen bonding. Those secondary structures, as they compact further, are going to make this molten globule. And then there's tweaking and turning of the side chains to reach to the tertiary structure. And this is sort of looking at, the good peptide and the bad peptide, right? So here we have this newly synthesized polypeptide. We'd like to think they all go into a native configuration. Many do. But suppose they misfold. Misfolded peptides are going to then undergo a number of alternatives at that point. A misfolded structure can, in some cases, be unfolded and then refolded and finally get to the native structure. If that doesn't happen, within not too long a time, it's more likely to then end up either being an aggregate or being degraded, being broken down by proteases. These aggregates are an important part of the diseases like Alzheimer's and things like that. So, did we miss? Let's see. I'm going to move on to heme because I think we're getting kind of late into the semester. So let's talk a little more specifically about the heme proteins, myoglobin and hemoglobin. This heme prosthetic group is found in a lot of the proteins. We're going to see more of them later on as well. Some of the proteins that it takes part in take advantage of this iron atom in the middle to do redox reactions, changing electron numbers, and we'll see those when we get into oxidative phosphorylation. And we know from its distribution that this is a very, very old molecule. Cells have been using heme for all kinds of interesting purposes for a long time. Myoglobin uses heme to do a fairly simple thing that's nonetheless pretty important. Myoglobin can bind oxygen reversibly. It can bind it and let it go, bind it and let it go. And it does so through the iron at the heme prosthetic group. So here's the oxygen atom, a molecule, so it's diatomic oxygen. It's binding at Fe2, so it's a particular oxidation state of the iron. And it can be, interacts with the histidine side chain nearby. The overall structure of the myoglobin holds that myoglobin in what's been called a hydrophobic pocket. When you look at the side chains around the myoglobin, they're all hydrophobic side chains. And so they exclude water from that area, and that iron is kept from rusting. Oxidation of Fe2 is F to Fe3 would rust it. If you look at, your car, the parts that are brownish red, that's Fe3. So the hydrophobic pocket is a specialized region within the myoglobin molecule that's essential for it to be able to do this. Now we can look at how oxygen is bound to myoglobin, and as you might expect intuitively, the more oxygen that's around, the more likely it is that the myoglobin will be occupied. And that's what this graph is showing you. As we raise the concentration of oxygen, the partial pressure of oxygen, TORs here, as we raise the concentration of oxygen, we're going to change saturation. Y is the fraction of the molecule that is saturated. And we can talk about a concentration of oxygen at which 50% of the myoglobin molecule is saturated. So again, with this business of half the site saturated. So we have a number, the P50 for myoglobin is 2.8 torr. It doesn't take much oxygen to half saturate a myoglobin molecule. That's going to turn out to be important for its role in sort of the oxygen economy of muscles. Hemoglobin is a little more complicated. Myoglobin is a single polypeptide. It has primary, secondary, tertiary structure. It can bind oxygen reversibly. Hemoglobin is a heterotetramer. That is, there are 4 polypeptides, and they're 2 each of 2 different ones. If it was all the same, it would be a homotetramer, but it's a heterotetramer. Now, the kinds of forces that can hold the subunits together are very similar to the kinds of forces that hold tertiary structures together. In general, when you see a protein that has quaternary structure, it's made up of multiple polypeptides, it's likely to have symmetry of one kind or another. So you can just see here some examples of, here's one with a trimer. These are, I don't know quite how many different subunits these guys have, but you can see they tend to have either mirror symmetry or rotational symmetry and so on. Having quaternary structure buys you a couple of different things. One, it's a way to get bigger. In general, things with quaternary structure are bigger than things with just tertiary structure. But it's also fairly common, not universal, but fairly common for quaternary structure to be associated with regulation. That having multiple subunits is part of the molecular machinery that allows a molecule to read the environment in one way or another and do different things under different circumstances. When we compare myoglobin and hemoglobin, they're both of them members of a family called globins, no surprise. And when we look at the globins, we can see something about the evolution of those molecules. All globin proteins are going to contain a heme molecule. They all have this hydrophobic pocket of hydrophobic chains that kind of keeps it from rusting. So you can see here the hydrophobic pocket like that, right? Hydrophobic pocket right around the heme group, the heme prosthetic group. In all the globins, there's a histidine on the F helix at the 8th position that can coordinate to the iron 2 that's sort of holding on to it from behind. And then on the 7 helix, there's a hydrogen bond from the histidine to the oxygen. So these 2 histidines are able to play a key role at holding on to the hemoglobin, heme group, and holding on to the oxygen. This is a superposition of 3 of these globins, of myoglobin and alpha globin, or alpha hemoglobin and beta hemoglobin. And you can see that the tertiary structure of these 3 is very, very similar. You can see how much you can overlay the alpha helix here, the alpha helices here, and so on. So the tertiary structures of all these globins is quite similar, which you'd think it would have to be to create that hydrophobic pocket, to put those hemes in the right place, and so on. Strikingly, their primary structure is quite different. Primary sequence is only 18% identical. So you can have this similar, a tertiary structure with very different primary sequence. And when we look at the different residues through here and the different places where there have been changes, we can say for any given residue, comparing across all the globins, either it's invariant, like the histidines, for example. If it's invariant, it's there in every one of them. It may be conserved, so there might be a valine or a leucine. There might be an asparagine or a glutamine. So in a conserved substitution, the general properties are similar, but there is some difference, some small difference between them, small difference in size and so on. Or it might be a position in which lots of things can occur, that you can find any of 18 different amino acids there, or even more, depending on the kind of domain we're talking about. So there's looking across this primary sequence, we can learn things by looking at whether the residues are invariant, conserved, or variable. And when you, for instance, look at some examples of that, you can then deduce that how these proteins evolved over time and how they're related to each other. And so if you look in here, here's, we're again comparing myoglobin, hemoglobin alpha, and hemoglobin beta. They're showing you the different helices, the A helix, the B helix, and so on. And you can see here's a leucine that's conserved among all of them. Here's a string of 4 residues that's conserved among all of them. So those are ones that are invariant across them. And then you can see some others which are being conserved between the hemoglobins. They're different than the myoglobins, but they seem to be important for hemoglobin structure. And then you can see some others where it can be any amino acid or even nothing at all. Here's a gap where that hemoglobin actually has fewer residues than the other ones. So these places where the amino acids have been kept the same are usually clues to what the overall structure is and how it forms. They're usually limits of what can or can't be changed. We looked a little bit at how myoglobin interacts with oxygen. Hemoglobin is more complicated, and that's one of the reasons it can do what it needs to do. Hemoglobin is a tetramer. I mentioned tetramers tend to go along with coordinated or regulated behavior. And this graph shows you the contrast in the behavior of the 2 different molecules. So this dotted curve here is the myoglobin curve, showing you here the P50 of a few tors up in there. It's also a hyperbolic curve, a simple-looking curve, if you like, one that you probably recognize from high school math. Here on the contrast is the curve for hemoglobin binding. And so if you sort of look at it qualitatively, as you raise the concentration of oxygen, so here's the oxygen concentration. As the oxygen starts to rise, hemoglobin starts binding some oxygen, but it's not ripping into it like the myoglobin. And then the binding picks up. There's an inflection point in there, and then it peaks out and stabilizes close to saturation. What's significant about this? Okay, so one of the things you can do is ask, where is the hemoglobin half saturated? You can see that the hemoglobin is half saturated at a much higher concentration of oxygen than myoglobin is. That's one important thing. It has a lower affinity or a larger P50 for oxygen. But the second is this sigmoidal shape of it all. And what this does for us is several-fold. In the lungs, where there's an abundant concentration of oxygen, the hemoglobin can be fully saturated with oxygen. Hemoglobin, of course, is inside red blood cells. It's not traveling as a free protein. It's in the red blood cells. And the oxygen in the lungs will completely saturate that hemoglobin so it's all occupied. It circulates through your bloodstream and moves on down, and now it's down out into the tissues. Well, the concentration of oxygen in the tissues is quite a bit lower. And the reason this blue band is so wide is because the oxygen concentration in the tissues will vary with what you're doing. If you're running fast, you're going to be using up your oxygen. The concentration of oxygen will be lower. If you're just lolling around watching the football game on TV, oxygen concentration will probably be higher. And so what you see in here is that the lower the concentration of oxygen in the tissues, the more completely the hemoglobin releases the oxygen that it's carrying. The myoglobin is fairly well saturated, but you can get almost a twofold difference in the extent to which the hemoglobin dumps its oxygen and unloads it into the tissues. And as it unloads it into the tissues, the myoglobin picks it up and is making it available for the uses of the cell. So the myoglobin is acting kind of like a buffer of oxygen, temporary storage between the bloodstream and whatever the cell is going to use it for. Questions about this before we go on? Okay. All right. Now that's a very cute trick. How does hemoglobin pull it off? Well, it actually changes shape. And this is one diagram of what some of those changes look like. You can think of hemoglobin as being made of two dimers, an alpha-beta dimer and a second alpha-beta dimer. And they twist relative to each other depending on whether the oxygen is present or not. And so if you look at deoxyhemoglobin with a relatively large cavity, if all the four, so this is, remember this is like 4 copies of myoglobin. There's 4 polypeptides here. There's 4 heme groups here. Each of those heme groups can bind an oxygen. In this particular configuration, none of them have oxygen. But if all 4 of those are saturated, then we talk about oxyhemoglobin. And oxyhemoglobin is related to deoxyhemoglobin by this kind of a twist, kind of like those magic boxes you get at Vortex Gem. So the alpha-beta dimer kind of twists relative to the other one. Notice here at the center. So each of these is like a myoglobin tertiary structure. Each of them has its own hydrophobic pocket where the heme group is. But because hemoglobin is a tetramer, there's the possibility of additional structures, and there's this central cavity now that's formed between the 4 subunits. And in going from deoxy to oxy, this central cavity has gotten smaller. So that's one of the hallmarks of the change in structure between the deoxy and the oxy state. One of the more visible ones, maybe. Now, what's interesting about that is that as the heme is twisting and changing, the heme rings from one subunit are talking to heme rings in other subunits. The heme groups within the individual polypeptides can actually communicate mechanically. No telepathy, just simple mechanical communication. And this is showing you the 2 possible configurations. That in the deoxyhemoglobin, the heme group is a little bit puckered. And then when the oxygen is bound, so here you can see an oxygen bound, then the heme pulls on the histidine on F8, and then that pulls on the F helix, and that twists the dimer, alpha-beta dimer, and the central cavity becomes smaller. And so there are some intermediate states that we're not going to explore now, but there's 2 sort of general states to think about that have been called T and R. So T for a tense state and R for a relaxed state. And in binding oxygen or letting go of oxygen, it goes between these 2 states. But this kind of mechanical crosstalk means that the binding of oxygen to one heme group on one subunit can influence the availability of a heme group somewhere else. This is showing you a change in some of these contacts. So I remember I talked about this twist. There are actual side chains that touch each other in different configurations. So in one configuration you can see here, this is a histidine that's kind of resting in between two side chains, a threonine and a proline. And as it changes its oxygenation state, this histidine switches over to the adjacent little valley. So it's really kind of like, you know, when you zip a zipper one way versus another way. So the tense state has different contacts between the subunits than the relaxed state. Just to get a little bit more complicated and to increase the utility of hemoglobin, the oxygen affinity of hemoglobin is also influenced by protons. And a deoxyhemoglobin, so over here we're in the tissues here. So here's a deoxyhemoglobin molecule that doesn't have a hemoglobin bound. It's capable of picking up binding protons and carrying that with them as they're making their motions out from the tissues to the lungs. And this binding is to some extent either or. In other words, that when you have an oxyhemoglobin, if it's going to bind a proton, that's going to stimulate it to have released that oxygen molecule. So the hemoglobin is acting to carry oxygen from the lungs to the tissues, but it's also acting to carry protons back out to the lungs. Now you think about your muscles when they're working hard, they can generate lactic acid and other acids. They're going to generate carbon dioxide from oxidative phosphorylation. And so the proton that hemoglobin picked up is because the carbon dioxide that was produced by the energy-producing processes in the lungs, and the legs rather, or the limbs, the periphery, if that carbon dioxide became hydrated into carbonic acid, the carbonic acid lost a proton that was picked up by the hemoglobin. The bicarbonate itself comes along in the bloodstream. You get to the lungs, and in the lungs there's lots of oxygen, so the oxygen knocks the protons off of the hemoglobin. The protons are now available to rebind to the bicarbonate ion and can break back down into carbon dioxide. So the hemoglobin is doing a second thing. It's helping to get the acid, the protons of the acid out. It's also helping to get the actual carbon dioxide out because it's carrying the protons it'll need to reform it in the periphery. So what this means is that the affinity of hemoglobin for oxygen will be affected by the pH of its environment. And that will enhance its ability to serve as a good carrier of oxygen to the places that need it. I think the next graph shows this as an affinity curve. The difference in these different curves, the fact that the different hemoglobins are showing different behavior at different pHs, is called the Bohr effect. And so in the most acid conditions, 6.8 is the most acid of these conditions. It has the highest concentration of protons. It's going to have the most protons to bump oxygen off with. It has the lowest oxygen binding out at the periphery. It has the largest P50. It has the lowest affinity for oxygen. Excuse me. When if you compare a rather alkaline pH here, you can see that the curve of the hemoglobin now is resembling myoglobin more closely, and it's not going to dump off as much of its oxygen in the periphery, and so it's not going to carry carbon dioxide out to the lungs, and it'll stay behind and help to restore the pH of the periphery. So, any questions about that before we go on? hemoglobin has been a purified protein for many years, and it has been the basis for a lot of structural studies. Understanding hemoglobin has given us insight into how proteins work in a lot of different ways. And I think hemoglobin S is one very valuable example of how that works. Hemoglobin S is a variant of hemoglobin that's named for the fact that it is able to confer sickle cell trait on the people who carry it. And whether it's a good or a bad gene depends on how many copies of it you have and where you live. In individuals who have one copy of hemoglobin S and one copy of hemoglobin A, their red blood cells will be a mixture of hemoglobin A and hemoglobin S. And those red blood cells will be relatively protected against malaria. And so when we look at populations of human beings where hemoglobin S is first observed, they tend to be along the malaria belt. There's a drawback to hemoglobin S, which is if you have two copies of hemoglobin S, under stress, your red blood cells can do this, that the modified hemoglobin is no longer fully in solution and instead makes the red blood cells curl up and sickle like this. And what's amazing about hemoglobin S is how small the change is. Hemoglobin S is the result of a single amino acid substitution. One amino acid on the surface of hemoglobin is altered, and that accounts for the entire phenotype. The resistance to malaria, the sickling, the damage to the lung, to the organs if you have too many episodes of it, and so on. So here we have an example of how important structure can be. One single residue on the surface of hemoglobin accounts for the whole phenotype. In very malarial areas of the world, heterozygotes might be 25 to 30% of the population. So it can confer a considerable beneficial effect if you have the pressure of the malaria around. Questions about, any other questions about hemoglobin S? Okay, let's move on then. Another aspect of regulation has to do with that cavity, that pocket in the middle that I tried to draw your attention to in deoxyhemoglobin. Remember that deoxyhemoglobin has a larger hole there and oxyhemoglobin has a smaller hole. In that central cavity, there can be bound a small molecule called bisphosphoglycerate, or BPG. So this central cavity of hemoglobin can interact there with side chains and be held there stably. And this is a little diagram showing you of how bisphosphoglycerate interacts there. If you listen to that bisphosphoglycerate, so a phosphate group is going to confer negative charges on BPG. So BPG, which you can see down here, here's negative charges and so on. Quite a negatively charged little molecule. We're going to see BPG again when we get into glycolysis. And it's interacting with side chains that are very positively charged. So here's a histidine and a lysine and a histidine and a histidine and a lysine and a histidine. So those positive charges there interact with the negative charge of BPG to hold it there stably. So it acts like a doorstop. When BPG is there in the middle of deoxyhemoglobin, it's like a little doorstop that resists that twisting motion to get to the oxyhemoglobin. It's the presence of BPG in that cavity that contributes to the relatively low affinity of completely deoxyhemoglobin. And the difference is quite striking. So this is showing you the curves of hemoglobin with BPG and without BPG. And you can see that in the presence of bisphosphoglycerate, the hemoglobin has this strongly sigmoidal curve that has this beneficial binding in the tissues so that it can give a graded response of dumping its oxygen in the tissues. In the absence of BPG, it's not quite as the same as myoglobin, but it's more like it than we would like it to be. It's not nearly as functional. And discovering the requirement for this was one of the contributions that Iowa made to blood banking. It wasn't unpacked instantly, but the discovery that red blood cells need glucose to stay healthy was made here, and it extended the life of blood from a few days to a month and a half or something. And a good chunk of that is due to the fact that red blood cells have to make BPG in order to keep the hemoglobin responsive to different concentrations of oxygen. Another place where these differences in hemoglobin affinity turns up is in fetal hemoglobin, that when you look at the red blood cells of a fetus, its hemoglobin has a higher affinity for oxygen than the mom's does. And so in that circulation, there's a transfer of oxygen from the mom to the fetus. Any pregnant woman who's 6 months pregnant or more who goes to the mountains knows how good the affinity of fetal hemoglobin is for oxygen. I've done it. This is another way of representing deoxyhemoglobin, and I think you can see here very clearly that BPG bound there in the center, acting like a little doorstop to reduce the affinity of the oxygen, which is a good thing in this case. You can also pick out, I think, the different heme rings. There's one of them. There's another one. I think, where are the other heme rings? There's another heme ring. Another one over there somewhere. Another way of looking at proteins that makes it a little easier to see kind of the large-scale features is sometimes called a sausage diagram. And here you can see, again, that there's 4 subunits to a hemoglobin molecule. Here I think you can see a little more clearly the heme groups that are in each subunit. So each of the 4 subunits has its own heme group. Each of them has its own hydrophobic pocket. And then here in the center is that BPG pocket that can modulate the affinity for oxygen. This representation is just trying to get across to you this notion of hemoglobin as a dimer of dimers, that an alpha-beta dimer rotates as a pair over the other dimer. The heme ring itself is not just a piece of chicken wire. The exact structure of the heme ring is important to whether or not it's going to bind oxygen or not. And this is showing you just how that heme ring is actually capable of twisting and bending in a number of different ways. The remarkable flexibility and variety of heme is part of why we're going to see it again and again as we go down through the rest of the semester. This is another way of representing the exchanges that are going on. Here again, we're looking from the lungs to the tissues. In the lungs, as you have the oxygen taken up, and you're going to have a fully oxygenated hemoglobin in the arteries, dropping off into the tissues to go over to the myoglobin. And then in the tissues, picking up carbon dioxide to be carried along, in some cases actually on the amino terminus of the hemoglobin molecule itself. That is, the amino terminus can bind onto that. That's showing you that in a little more detail. Have a good weekend. See you all on Monday. AV? AV? Yoo-hoo! Sorry.
Oh, that's all right. Just trying to get down the stairs.
Yeah, so I think we have, so we did get most of the lecture then?
Oh, yeah, it was going.